Good afternoon, everyone, and welcome to another webinar sponsored by the North American Vascular Biology Organization. I'm Enar Cuervo from the University of Illinois at Chicago and a member of the NAVBO Education Committee. I will be moderating today's session. We are pleased to welcome our speaker, Sathish Srinivasan. He's an associate member at the Oklahoma Medical Research Foundation in Oklahoma City. He will present his research entitled Wnt Beta-Catenin Signaling, Mechanotransduction and Lymphatic Vascular Development. At this time, I'd also like to welcome Boksik Cha, also from the Oklahoma Medical Research Foundation. He will be monitoring today's question. The way we handle question is in two different ways. Throughout the presentation, you can type your questions into the question box of the control panel. These questions that you type during the presentation will be answered at the end of the presentation. Dr. Cha will compile them and then pose this question to Dr. Srinivasan at the end. Once we've gone through all these written questions, there's going to be, provided that there's time, the ability to ask additional questions live in real time. So if you still have a question, you can raise your hand by clicking on the hand icon of the left side of your control panel. Then you will be recognized, I will call your name, your mic will be unmuted, and you will be able to ask your question live. If you experience any technical problems throughout the webinar, please click the Help tab at the top of the control panel. Then scroll to the bottom of the Help screen for the GoToWebinar technical support phone number. This webinar is being recorded and archived on the NAVBO website for future use. Dr. Srinivasan received a PhD degree from Tulane University in 2004. He trained as a postdoctoral fellow in the lab of Dr. Guillermo Oliver, a pioneer in the field of lymphatic vascular development. He joined the Oklahoma Medical Research Foundation in 2013, where he is currently an associate member at the Cardiovascular Biology Research Program. His lab is currently focused in developing approaches to gain a clearer understanding of lymphatic vascular development, generating mouse models of human lymphedema, and identifying the anatomical, developmental, and mechanistic defects of these mice, and developing innovative treatments for lymphedema and the associated cardiovascular disorders. He has made significant contributions to science. Some of them are the demonstration of the venous endothelial cells as the major source of lymphatic vasculature, the discovery of CUBTF2 as the activator of PROX1 expression, the identification of a positive feedback loop between PROX1 and VEGF receptor 3. He's involved in revealing the stepwise morphogenesis of lymphovenous valves, which regulate lymph return to blood circulation, and the identification of Wnt beta-catenin signaling as the regulator of lymphatic vessel maturation and valve development. Today's webinar will present how the recent discovery of Wnt beta-catenin signaling is a critical regulator of lymphatic vascular development, and also the role of shear stress and its relationship with Wnt beta-catenin signaling in the lymphatics. Welcome, Dr. Srinivasan.
Thank you, Enar, and thank you all for joining this webinar. I'm going to, let's first of all remove this embarrassing picture. Let's go and to the title of this seminar. So we are going to be discussing about Wnt beta-catenin signaling, mechanotransduction and lymphatic vascular development. I am joined here by Boksik Cha. He's a research assistant member here at Oklahoma Medical Research Foundation. And he was the main guy who basically brought my lab into this field. And please ask any questions that you have. And if I cannot answer the question, Boksik will definitely be able to answer it. Thank you again. So a brief introduction to lymphatic vasculature. So blood vasculature, as you all know, carries nutrients like oxygen and glucose to every cell of the body. And due to differences in the protein concentration and the blood pressure, fluids constantly leak from the blood vasculature into the interstitial space. And the main function of the lymphatic vasculature is to reabsorb all this fluid and return it back to blood circulation. The lymphatic vasculature also plays at least two additional roles in mammalian physiology. And one is to function as a route for immune cell trafficking. So if there is an infection in any part of the body, the dendritic cells, antigen-presenting cells, they all recognize those foreign particles. They enter into the lymphatic vessels, which are loosely attached to each other. The cells are loosely attached to each other. So it provides an easy route for those immune cells to enter. And from there, they go to the lymph nodes where they trigger the humoral response. So, and also in the gut, lacteals are specialized lymphatic capillaries that function to absorb digested lipids. Large lipid particles are taken up by these lacteals, which then take them to blood circulation. So, and the lymphatic vessels are present throughout the body. This is the architecture of the lymphatics. You have the capillaries, also known as the initial lymphatic sinus, which are the place where the fluid, interstitial fluid, enters into the lymphatic vessels. And from there, they are transported into the collecting vessels, from which it goes on to the thoracic duct and then return to blood circulation. And there are valves present within the collecting vessels, which regulate the unidirectional flow of lymph, and also prevents regurgitation, reflux of fluid. And one of the very interesting aspects of the lymphatic vasculature is that although the lymphatic vessels are present throughout the body, these major lymphatic vessels, the thoracic duct on the left side, and the right thoracic duct, or right thoracic vessel on the right side, they converge into the blood circulation, merge with the blood circulation at the junction of internal jugular vein, subclavian vein, and superior vena cava. And this is just above the heart. So the idea is, because the pressure within lymphatic vessels is really low, the low pressure that is caused by the pumping action of the heart during the diastole creates a vacuum, and that is able to absorb the lymph fluid from the thoracic duct. Okay, so there are two pairs of lymphovenous valves located at this junction, and we will be talking about it quite a bit in this seminar. So what happens if the lymphatic vasculature is defective, right? And it leads to a disease known as lymphedema. And here are some of the individuals. You can see that they have swollen legs. And that is due to the accumulation of fluid within the interstitial space. So some of the patients may develop unilateral lymphedema, meaning one of the legs or one of the hands could be swollen. But in other cases, you can have swelling in both the legs, both the limbs. And an additional consequence of lymphedema is inflammation. So here in this panel B, you can notice that the tissue is inflamed, it's red in color. And also another consequence is infection, because the lymphatic vessels are not functioning properly. They could develop papillomas, they are prone to infection, and you can see that the nail is damaged in this particular individual. Likewise, in panel B also, you can see that this person has papillomas in the foot. The mutations in genes could cause hereditary lymphedema. It's also known as primary lymphedema, and that is what my lab is working on. We are interested in understanding the role of these genes which regulate, mutations in which cause lymphedema. And this primary or hereditary lymphedema could further be classified based on the onset, the time of onset of the disease, into congenital lymphedema, which happens immediately after birth, or it could also happen in utero in fetuses. And And also it could be called as lymphedema precox, which happens sometime after birth, but before the age of 35. And finally, there is lymphedema tarda, which happens after age 35. And there is quite a bit of overlap between these this classification. Mutation in certain genes could cause congenital lymphedema, but it could also cause lymphedema precox, or mutations in certain genes could cause lymphedema precox and also lymphedema tarda. So, so there is quite a bit of, it's a loose classification. And according to this website, rarediseases.org, approximately 1 in 6,000 individuals develop primary lymphedema. So it's a rare disease, but it's not so rare considering the population, human population throughout the world. So it's quite important that we understand how these genes are functioning, what are they doing, what exactly is happening in these individuals, and what are the physiological consequences that result from lymphedema. Okay, so and lymphedema, congenital lymphedema, hereditary lymphedema could also be further classified based on the associated disorders. So some genes, mutations in these genes could cause just lymphedema. So as an example, if this is isolated, it's called isolated lymphedema. An example is mutations in this receptor tyrosine kinase, VEGFR3, or its ligand, VEGF-C. And if a mutation in a gene causes various disorders, and lymphedema being just one of them, it's considered a syndromic lymphedema. So for example, mutations in FOXC2 could cause lymphedema. It could cause distichiasis. This is double rows of hair, this hair in the eyelashes. They can also have yellow nails, etc., etc. Okay. And I have underlined some of the genes that we have been working on in my lab. We have published some papers, we have collaborated with others. I encourage you to have a look at those publications if you have a chance. Okay, so what do these genes do, right? So to understand the mechanisms is very important, as I said. So what do genes do? They either regulate the formation of structures like the lymphatic vessels, or they regulate the functioning of the lymphatic vasculature. Now, to understand the role of genes in functioning, you will need a webinar by someone like Mike Davis at University of Missouri, Columbia, who's an expert in understanding the mechanisms of lymphatic function. But what I will be talking about is about the role of genes in the development of the lymphatic vasculature. So we'll start from the very beginning. So the lymphatic vasculature, this is in mouse embryos. They start to develop at approximately embryonic day 9 in mouse embryos. And subpopulation of venous endothelial cells, they turn on the expression of this transcription factor called SOX18 in a polarized manner. And SOX18 cooperates with another transcription factor called CoopTF2, which they both appear to cooperate and activate the expression of PROX1, which is a transcription factor that is considered as a master gene of lymphatic vascular development. And PROX1 was reported by my mentor, Dr. Guillermo Oliver, when he was at St. Children's Hospital. And I reported the role of CoopTF2 while I was a postdoc with Guillermo. And SOX18 was reported by Matt Francois, who is in Australia. Now, once PROX1 is turned on, those cells then, they are known as the lymphatic endothelial progenitors on the vein. And these cells, they migrate out of the vein under the influence of VEGF-C, vascular endothelial growth factor C. And VEGFR3 is a receptor for VEGF-C. PROX1 regulates the expression of VEGFR3. And the migrating cells, they turn on genes such as photoplanin, and at this stage, we call them as mature lymphatic endothelial cells. And these cells, they coalesce to form the lymph sac, the primitive lymph sac. And from the lymph sac, again, lymphatic endothelial cells sprout, migrate throughout the body, and they form the lymphatic plexus. So this is the first step of lymphatic vascular development. The second step of lymphatic vascular development is also very critical. So this lymphatic plexus that I mentioned to you previously, they further undergo maturation to form the capillaries that I mentioned to you about previously. So the capillaries have these button-like junctions, meaning they have a lot of gaps in between to allow fluid entry into the lymphatic vessels. And then they also, these collecting vessels, they also undergo maturation. They form zipper-like junctions. They are very tightly associated with each other to prevent leakage. And then the valves also develop within the collecting lymphatic vessels. Okay, so various signaling molecules, have been reported to play a role in the development, either the first step of the lymphatic vascular development, which I mentioned, PROX1, VEGF-C, VEGFR3, COOP-TF2, SOX18. But there are other signaling molecules that have been reported to regulate the maturation and valve formation of the after the primitive plexus, lymphatic plexus forms. And this includes integrins. You have integrin alpha 9 reported by Tahia Mechanotransduction. You have integrin alpha 5 reported by Christopher Turner. Ephren B2, Ephren B4. Again, Tahia Mechanotransduction has done important work on this pathway. Then adrenomedullin is another important molecule that was reported by Kathleen Caron. It's important for the formation of cell junctions. BMP9 appears to play a role in valve formation and maturation. Neuropilin signaling reported by Anne Eichmann. They are very important. And then finally, Notch pathway is also very critical for lymphatic maturation. And it's reported by Jan Kitaisky and group by several publications. And Tatsume Kume also made some publications on that. So one particular molecule that seems to play a very important role in lymphatic vascular maturation is this transcription factor called FOXC2. And Tahia Mechanotransduction, Tatiana Petrova in Switzerland, she has made some very important contributions about FOXC2. And here you can see in a control embryo, the lymphatic vessel is undergoing maturation by downregulating the expression of VEGFR3. Then the valves are forming within those mesenteric lymphatic vessels. But if the embryos are deleted of FOXC2, you can see that the vessels are immature. VEGFR3 remains really high, which is a sign of immaturity of the lymphatic vessels. The valves are missing. And also, if you look at the capillaries in panels D and C, the lymphatic capillary of a control embryo is devoid of smooth muscle cells, which allows easy entry of fluid. But the mutant capillaries, they have abnormal alpha smooth muscle actin positive cells accumulated. It prevents function. So obviously the question is, if FOXC2 is a master regulator, or it seems like a very important regulator of lymphatic maturation, how is FOXC2 regulated? What are the pathways that are regulating the expression of FOXC2? And what are the targets of FOXC2 has been an important focus in the field by many labs, including my own. And as I mentioned several signaling pathways that are important for the maturation of lymphatic vasculature, but none of those signaling pathways appear to have a phenotype that is similar to that of was similar to the FOXC2 nerve embryo. So again, here is a publication from Tatiana Petrova's group, and they came up with this really beautiful model to explain the maturation of the lymphatic vessels. So as I mentioned to you previously, the lymphatic vessels do not have that much of pressure within. So in initial immature collecting vessels, there is this oscillatory flow. The lymph flows back and forth within the vessel. And eventually, the idea is that the valves develop at those sites of oscillatory flow, eventually resulting in the flow getting streamlined into laminar flow that prevents the backflow of lymph. But on the sinus side of the valve, you still have this reflex going on. And the idea that Emily Sabine and Tatiana Petrova presented in this work is that this oscillatory flow itself is very important for the expression of FOXC2 and ultimately the maturation of lymphatic vessels. And in support of that idea, they showed this very important data. They exposed lymphatic endothelial cells to oscillatory flow, which is OFS here. And they showed that FOXC2 expression is indeed upregulated by this OFS. In contrast, laminar shear stress either doesn't regulate FOXC2 expression, or if anything, maybe down-regulated a little bit. So the idea from this work is that the oscillatory flow, the lymph flow, it activates the expression of FOXC2, which in turn activates downstream pathway to regulate lymphatic valve formation and lymphatic vessel maturation. But the question is, how is this lymph flow sensed by the lymphatic endothelial cells? So what is in between the cells at the flow on the cell surface, and how is it translated into FOXC2 expression? And we got into this question almost by accident, a fortunate accident, I should say. So as I mentioned to you, many of the signaling pathways don't appear to be exactly recapitulating the FOXC2 phenotype. So my lab was interested in screening for other signaling pathways to see whether they could play important roles in lymphatic development. And at that time, Boksik Shah, this handsome man, he joined my lab as a postdoctoral fellow. And he has a background in Wnt signaling pathways. His PhD was on cancer cells and cell lines. So he joined the lab with this strong ambition and interest. He wanted to know the role of Wnt signaling in development. And he said, because we don't know anything about Wnt signaling in lymphatics, I would like to explore whether Wnt signaling pathway plays any role in the lymphatics. So what exactly is Wnt signaling pathway? Wnt is one of the very important signaling pathways in any organism development. And it's very simple signaling pathway, in fact. So there are these receptors called frizzle, which are, and I believe there are 10 frizzle receptors in mammals. And these frizzle receptors are expressed on the cell surface. And LRP5 and 6, those are called the co-receptors for frizzles. And in the absence of Wnt ligand, there is this critical molecule called ß-Catenin. It remains either on the cell membrane attached to adherent junctions, tight junctions, or they remain in this complex known as the destruction complex, composed of actin, APC, and GSK3. So these complex, the destruction complex, keep degrading beta-catenin so that it cannot activate gene expression. However, when Wnt ligands, and there are 19 different Wnt ligands in mammals, when they associate with the receptor co-receptor complex, then by a really complicated mechanism, beta-catenin is now released from the degradation complex, and it goes to the nucleus, where it activates many molecules. Okay, so beta-catenin gives a really for a geneticist, it's a very convenient tool to check whether Wnt signaling is important or not. So, and one of the first experiments that Boxic did was use this transgenic mouse line. And here we have a GFP expression, H2B is nuclear GFP, expressed under the control of TCF binding sites. So TCFs are the transcription factors to which beta-catenin associates with. When Boxic looked at the expression of GFP in these mouse embryos, we saw strong expression in the valve, developing lymphatic valve forming cells. And he was also able to observe some GFP expression in the lymphatic endothelial cells. And secondly, we did in situ hybridization for the gene known as AXIN2, which is considered as one of the very critical targets of Wnt signaling. And this is a picture of the developing lymphovenous valves. And you can see that AXIN2 is strongly expressed in the developing lymphovenous valves. So that gave us the confidence to go ahead and check the role of Wnt in lymphatic development. So Boxic deleted beta-catenin in the lymphatic vasculature using live-on-3. It's reasonably lymphatic specific. It is also expressed in some specialized blood vessel vascular beds. But it's a nice tool to investigate the role of genes in lymphatic development. And he found strong edema. You can see the swelling on the back of these embryos. They have edema. And when he analyzed these embryos further, he observed that they lacked valves, the lymphatic valves. It is in panel J. And the vessels also appear immature. So the expression of VEGFR3 is very high. And these are phenotypes which are very similar. I won't say identical, but quite similar to FOXC2 null embryos. So then he went on to check whether there is in fact any relationship between Wnt signaling and FOXC2 expression and the oscillatory shear stress model, which was an exciting model that was available at the time. So you can see that oscillatory shear, in fact, increased. This is in panel B. You can see that oscillatory shear increased the overall expression of beta-catenin. It also increased the expression of FOXC2. And when Boxic did the Western blot, he could see it is not just the beta-catenin, but when we use the antibody that is specific for the nuclear beta-catenin, that is also increased by oscillatory shear stress. And of course, you can see that genes such as FOXC2 and GATA2, they are upregulated by oscillatory shear. Then Boxic tested whether Wnt signaling is indeed required for expression of FOXC2. And for that, he used this inhibitor called ICRT3, which prevents the interaction between nuclear beta-catenin and the TCF transcription factors. And you can see that ICRT3 inhibited the expression of FOXC2 and GATA2 by oscillatory shear. So the model, and this is also another experiment that he did. He treated the lymphatic endothelial cells not in the presence of oscillatory shear, but just in static conditions. He treated the cells with the bio, which is a Wnt agonist, And you can again see that FOXY2 expression is and GATA2 expression is upregulated by this Wnt signaling pathway. So oh, I keep missing slides. So here is another data. So as I mentioned to you, TCF transcription factors, they associate with defined regulatory element, which is shown here in panel A. And one such regulatory, very highly conserved regulatory element was found in the FOXY2 regulatory element. And Buxik tested whether ß-Catenin in fact associates with this site, TCF binding site, and indeed it does. So, and finally, this is a very important experiment that he did. So Wnt is regulating FOXY2 expression. So does it mean that FOXY2 could rescue the defects caused by the loss of Wnt? So here in panel G, you can see an embryo that is deleted of ß-Catenin. You can see the vessels are dilated, they lack the valve. And in panel H, we generated a new mouse model to conditionally overexpress FOXY2. So what Buxik did, he deleted ß-Catenin, but in turn, he was able to activate FOXY2 in those Ly1 positive cells. And you can, if you compare panels F and H, you can see that the defects associated with the loss of ß-Catenin significantly rescued by the overexpression of FOXY2. So it gave us confidence that FOXY2 is in fact downstream of Wnt signaling in the lymphatic vasculature. The rescue, by the way, is not complete. We were not able to rescue the formation of lymphovenous valves. We were not able to rescue the formation of lymphatic valves, but the vessel patterning was quite dramatically rescued. So based on these results, we proposed a very simple model that oscillatory shear stress activates Wnt signaling. Wnt in turn activates FOXY2 expression, which promotes the development of lymphatic vasculature. But like any good science, any good report, once you publish one paper, you have 100 different questions that you want to test, right? So, and we had several questions as well. So some of the questions are like, okay, the oscillatory shear stress appears to activate Wnt signaling in the lymphatics, but does it involve the canonical pathway that has been reported, in most of the literature? What I mean by that is, are there Wnt ligands involved in this pathway? What about receptors? Are the frizzled and LRP co-receptor, are they involved in this pathway? And are TCF, LEF transcription factors involved in this pathway? And importantly, this shear stress dependent FOXY2 expression, is it unique to lymphatic endothelial cells? And if so, why and how? What are the mechanisms? So these are the questions that Buxik then decided to address in a follow-up work. So first question, Wnt ligands. Are Wnt ligands important for the shear stress response in lymphatic endothelial cells? So Buxik exposed lymphatic endothelial cells to oscillatory shear at for various time points. And one of the readouts that you will get, if the Wnt ligands are involved, if Wnt ligands associate with this frizzled LRP complex, the first thing that will happen is the phosphorylation of LRP6. And you can see that once we expose the lymphatic endothelial cells to shear stress, there is indeed the phosphorylation of LRP, which indicates that more than likely Wnt ligands are involved in this process. So second, this is a culture system. So we are growing lymphatic endothelial cells just by themselves. What is there is media and the lymphatic endothelial cells, nothing else. So where are the ligands coming from? Either the Wnt ligand is coming from the media, or the lymphatic endothelial cells are producing it by themselves and regulating Wnt signaling in an autocrine manner. So here is another interesting detail about Wnt signaling. So there are 19 different Wnt ligands in mammals. But for us, helpful for investigators, there are two molecules that are extremely important for the secretion of Wnt ligands. One is a molecule known as porcupine, and what it does, it adds a lipid moiety to the Wnt ligand. And this addition of lipid moiety is extremely important for the secretion of the Wnt ligand from any cell. The second molecule is known as Wnt-less. It's a chaperone molecule that is critical again for the secretion of Wnt from the cell. So if we delete porcupine or inhibit porcupine, or delete Wnt-less or inhibit Wnt-less, we will be able to abolish the secretion of Wnt ligands from any cell. So that is what we used. So the first experiment Boxig did, he again performed this oscillatory shear stress experiment, but now he treated the cells with this chemical called LGK, which is an inhibitor of porcupine. So in a control condition, when he treats the cells with oscillatory shear, you can see AXIN2 is upregulated, you can see FOXY2 is upregulated, GATA2 is upregulated. But if we inhibit LGK, then all the expression of these genes are significantly abolished by this chemical. So it gave a really nice model. So we were very excited. We were like, okay, cool. So the lymphatic endothelial cells are producing their own Wnt ligand, and it's promoting the differentiation of lymphatic vasculature. So let's test this model. And we use this conditional mouse generated by Richard Lang. He made this flox allele for Wnt-less. Again, as I mentioned to you, Wnt-less is important for the secretion of all Wnt ligands. So we deleted Wnt-less using live-3. And it was a big disappointment for us. And you can see that there is no phenotype. These might survive. They have ability, the lymphovenous valves are fine. The lymphatic vessels are fine. So, but Boxig persisted. He wanted to know, oh, all right, even if the autocrine Wnt signaling is not all that critical for lymphatic development, maybe the Wnt ligands are coming from other sources which are compensating for the Wnt from LECs. So he made double knockout. He deleted Wnt-less in many tissues, and what he found was this particular phenotype. So he deleted Wnt-less from vascular smooth muscle cells using SM22-Cre together with the live-Cre. And we were fortunate with the genotyping approach for this cross. And when he did this experiment, then the phenotype was very similar to the loss of beta-catenin. So these might say lack the lymphovenous valves, the lymphatic vessels were immature, the lymphatic valves were absent. So it was a very nice result. So what about the receptors? Again, there were 10 frizzle receptors, so it is nearly impossible to delete all the frizzle receptors. However, there are only 2 co-receptors which play a very critical role in Wnt signaling, LRP5 and 6. So when Boxig deleted LRP5 and 6, again, we see a very strong phenotype. So the valves, the lymphovenous valves were defective. They're absent in these embryos. And finally, what about the TCF transcription factors? And this question is related to the second part of the second question that I have listed here. And that is, are lymphatic endothelial cells unique in this Wnt FOXY2 pathway? And if so, how and why? So, and the answer is yes, lymphatic endothelial cells are quite unique. So if you expose lymphatic endothelial cells to oscillatory shear, we have FOXC2 overexpressed. However, if you expose blood endothelial cells to shear stress, Wnt is activated, but FOXC2 is not. So what are the unique characteristics of lymphatic endothelial cells that promotes the expression of FOXC2 by oscillatory shear? And I mentioned to you earlier in the talk that PROX1, the transcription factor, is considered as a master gene for lymphatic vascular development. So if you don't have PROX1, the cells lose their lymphatic identity. So what Buxik did, he knocked down PROX1 expression in lymphatic endothelial cells and then exposed them to either shear stress or to Wnt ligand. And in both the cases, you can see in the absence of PROX1, FOXC2 is not upregulated. In controls, you can see it's upregulated by oscillatory shear and by Wnt ligand. But if you don't have PROX1, it does not happen. Then, so PROX1, as I mentioned, is a transcription factor. And beta-catenin is also a transcription factor, right? It goes into the nucleus, it associates with TCF transcription factors. It's a transcription co-activator, to be precise. So what Buxik did, and beta-catenin is known to associate with other transcription factors. I guess FOXO1 is one such transcription factor which could associate with beta-catenin. So Buxik tested whether PROX1 could also associate with beta-catenin, and indeed, when he did this co-IP experiment, PROX1 and beta-catenin are associating with each other. And then he did this reporter assay. He took top flash, which is a reporter for Wnt signaling, and if you express beta-catenin, you can see the reporter activity goes up like 20-fold. But when he adds PROX1, a small amount of PROX1 to this mix, then the activity is increased threefold. It goes up like 60-fold higher than baseline. So PROX1 appears to act in the Wnt signaling, promoting quite dramatically the Wnt signaling within the cell. So how is this happening? Again, these are transcription factors. So Buxik tested whether PROX1 could associate with the regulatory elements of FOXC2, the TCF-LEF site, and indeed, in the presence of an agonist, Wnt agonist bio, PROX1 association with its binding site is increased quite dramatically. And finally, what are the TCF transcription factors? So we did some RNA-seq experiments to look at the expression of different TCF factors, and TCF7-like-1, which is also known as TCF3, the most highly expressed TCF factors in the lymphatic endothelial cell. TCF-like-2 is also expressed here, although the other, there are 4 transcription factors, by the way. The other 2 don't seem to be expressed that much. Now, these TCF factors, they associate with beta-catenin through their N-terminus domain. And if you delete the N-terminus of these TCF factors, they function like dominant negatives. So they remain associated with co-repressors, and they don't switch on to become co-activators of transcription. And fortunately for us, TCF3 delta and mice were made by Brad Merrill at the University of Illinois, Chicago, and they had reported that these mice have severe edema, and they haven't investigated the lymphatics. But this phenotype looked quite similar to our beta-catenin loss of function mice. So they were kind enough to collaborate with us. They gave us some samples to analyze. And you can see here that the phenotypes, again, were almost identical. I don't want to be exaggerating. Let's say that it's very similar to FOXC2 null embryos. So here you can see the smooth muscle cells are abnormally recruited to the lymphatic vessels. They don't have the valves. This you can see in panels G and H. And Boxic also looked at the lymphovenous valves, and these were devoid of the lymphovenous valves. And again, coming back to the same experiment, Boxic expressed the dominant negative form of TCF3 into the lymphatic endothelial cells and exposed them to either oscillatory shear or Wnt ligand. And this dominant negative form of TCF3 was able to prevent the expression of FOXP2 by this stimuli. Okay, so together we came up with this model whereby oscillatory shear stress promotes Wnt secretion from the lymphatic endothelial cells. But this is not sufficient for the lymphatic development. There is this additional source of Wnt coming likely from vascular smooth muscle cells. There could be other sources as well, we don't know. But these Wnt ligands, they associate with Wnt receptor, co-receptor complex to activate ß-Catenin, nuclear ß-Catenin, which then associates with FOX1 to promote the expression of FOXP2, then regulate lymphatic vascular maturation and lymphatic valve formation. So this was Boxic's paper from last year. With that, I would like to acknowledge, of course, Boxic has been the driving force behind this project. He deserves all the credit for his work, and we just helped him. And Shin, she has done a lot of experiments in this work. She did scanning electron microscopy, which I didn't show. She has done in situ hybridization for this work. And in the upcoming NAVBO conference, I'm going to be talking about a different signaling pathway, which Shin has explored thoroughly, and we are very excited about it. So be sure to join us there. And Legion has been critical for maintaining the lab. He has done a lot of experiments for this project as well. And Yenchun is a new postdoc. He's doing very exciting new research. Riyaz Muhammad, he was a graduate student in the lab. He just graduated yesterday. He's Dr. Muhammad now. And his paper is in press. It's online now in development about the role of GATA2 in lymphatic vascular development. I encourage you to go and have a look to get a chance. And I also want to thank a lot of collaborators throughout the year. They have been very kind to help us with many experiments, many reagents and ideas. Yang Kuan Hong and Dongwon Choi from University of Southern California, Dr. Mike Davis at University of Missouri, Hong Chen from Harvard, Brandon Dixon from Georgia Institute of Technology. I also want to thank my colleagues here at Oklahoma Medical Research Foundation, Courtney Griffin, Legion Shah, Lauren Olson. They have been phenomenal supporters and collaborators throughout the year. So with that, I thank you again for your attention and for joining us in this webinar, and I'll be happy to answer any questions that you may have.
Thank you so much, Dr. Srinivasan. That was fantastic work. I have a couple of questions that I'm going to ask while we gather the questions from the audience. And being a mural cell biologist, my questions are regarding the smooth muscle cells and the role that they may play in your model. So the first one is, why do you get recruitment of smooth muscle cells to the vessels when you have this defect on the FOX2C signaling? And the second one is, you've nicely proven that there is contribution from both the lymphatic and the smooth muscle cells of Wnt signalins in your model. But have you done the experiment only with smooth muscle cells? Is it possible that smooth muscle cells are the only contributor? It may be published in your paper, but I haven't had the privilege to see that control.
Oh, okay. Very good question, Senna. So the first question is why this mural cells, they are now called as lymphatic muscle cells because they have unique property in a normal mouse embryo, let's say. So why are they abnormally recruited to the FOXP2 embryos? And this work again was done by Tatiana Petrova in their characterization, and they reported that the signaling pathways that normally recruit vascular smooth muscle cells to blood vessels, and I believe one of them was ANG2 and TGF-beta signaling pathways. They are abnormally upregulated in the lymphatic endothelial cells of the proxy to null embryos. So this is a relatively old finding. This was from 2007 or something like that. There hasn't been many follow-up studies, but it is possible. It is also possible that because these capillaries are defective, they have too much of fluid accumulated. Maybe they are mechanically challenged as well. Maybe to prevent these vessels from exploding, we somehow develop an approach, maybe through the signaling pathways, to recruit these supporting cells. Now, to answer the second question, yes, we deleted Wnt plus only in the vascular smooth muscle cells with the SM22. It does have a phenotype. There are the vessels do appear abnormal. However, the valves, the lymphovenous valves that I was talking to you about, were more or less fine. So the live Cre mediated deletion of Wnt plus also had normal valves, and SM22 Cre also had normal valves. But when you do the double deletion, they were gone. So that is why we reported that we have two different sources for the development of the vasculature. Did I answer that?
Yeah, you did. Thank you so much. At this point, we have several questions from the audience, so I'm going to turn this over to Dr. Cha, and he can direct these questions to you. Thank you.
Okay. Okay, can you read it, book?
Okay, so we have one question that, so in our mouse model, besides the LVB defect, what is another defect? Do you have can you explain a little bit more detail? What is like a phenotype of the ß-Catenin null mouse or other Wnt inactivation mouse?
Okay, good question. Yeah.
So yes, they do lack lymphovenous valves. They lack the lymphatic valves of the collecting vessels, and the capillaries are also defective. They recruit the vascular smooth muscle cells abnormally to the vessels. And let me see if I show this picture. Yeah. So in panel G, you can see that the vessels are dilated. They are not able to migrate properly to the midline. So they are assume that the vessels are coming from both the sides and they are trying to reach the midline. But when you delete ß-Catenin, they are not able to do that. So yes, so they lack lymphovenous valves, they lack lymphatic valves, the vessels are dilated, and there is hypoplasia. And this live Cre is also expressed in venous endothelium. I should have mentioned this here. And within the venous endothelium, there are the venous valves, which are the first venous valves to develop. And when you delete ß-Catenin, those venous valves also fail to form. So the valves, all kinds of valves, the lymphatic valve, lymphovenous valves, and venous valves, they share a lot of similarities. So when you delete ß-Catenin, the venous valves also do not form.
Yeah, did I answer that question?
Okay, so the second question is, I think we discussed a lot of these subjects. So what is the OSS sensor? So what is the mechanism to sensing the shear?
Okay. That's a very good question as well. So there are a few sensors. Okay. ß-Catenin, as you may recall, Dr. Martin Schwartz from Yale, he reported several years ago that ß-Catenin is in this mechanosensory complex in the endothelial cells, which are very critical for mechanosensation. And recently, we collaborated with Joshua Scallon in University of Southern California, and we have shown that ß-Catenin is indeed very critical for mechanosensation and even Wnt transduction in lymphatic endothelial cells. And another molecule which is coming up, which was reported by my collaborator, Yang-Kwan Hong, from University of Southern California, is piezo-1. It's one of the very important touch sensors in many cell types. And when they delete piezo-1, again, there are several defects, including mechanosensation. So the cells are unable to now activate FOXY2 expression in response to oscillatory shear stress. So those are two molecules that are definitely important. But I will not be surprised if other molecules such as integrins or any tight junction molecules, they come in picture at from future studies. Okay.
Okay, so the other question is VEGFR3 expression is regulated by the Wnt signaling or FOXY2?
Okay.
So
very good question. The relationship between FOXY2 and VEGFR3 is quite complex. So when Tatiana deleted FOXY2 in lymphatics, it's a global knockout mouse, they were able to observe increased expression of VEGFR3 in the lymphatics, and also their localization appeared defective. So they seemed aggregated within the cell, and we have noticed it as well. With respect to Wnt, we definitely see increased expression of VEGFR3, but as I recall, we didn't really notice this abnormal localization, but we should, I think, look at it a little more carefully. So this suggested that maybe FOXY2 is like an inhibitor of VEGFR3 expression, which will correlate with the maturation process, right? The vessels, they downregulate VEGFR3 as they undergo maturation. But when they bred FOXY2 with VEGFR3 and they made double heterozygous mice, it actually made the phenotypes worse. They had, the severity, so the heterozygous FOXY2 heterozygous mice had milder defects, but when they made the double heterozygous mice, the severity, it was so severe that it looked almost like FOXY2 null embryos. So the relationship between VEGFR3 and FOXY2 is a little complicated. Just like anything else in biology, there could be feedback and things like that. But it's an important question. It has to be explored more thoroughly in the future.
Okay, so this is gonna be last question. That do you know which result are important for the lymphatic vascular development, and also the Wnt5b probably?
sorry, what?
Wnt5b.
Wnt5b.
The Wnt5b is important. So frizzled, we don't know. We did see several frizzled receptors coming up in our RNA-seq. We don't have the courage to delete all the different frizzles in the lymphatics. Wnt5b, trying to remember, I think Mark Kahn published a paper in Developmental Cell showing that Wnt5b is important for heart valve development. The 5A, beta, BD. So okay, so the what?
Is a publication about 5B.
Okay.
Yeah.
Yeah.
So,
Oh, I see. It's a non-canonical.
Yeah,
non-canonical. Yes.
So we don't really know the 5B is really important.
Okay. Yeah. So Buxik is saying that 5A, and I remember this publication from George Welting's group in Germany. They showed that Wnt5A is important for lymphatic development. It's a non-canonical Wnt, by the way. It doesn't activate ß-Catenin, but it activates, the planar cell polarity, cytoskeleton, things like that. We did not check whether we don't have those finds, and we did see Wnt5B, and they have a milder defect. Is that right, Boksit? Can you remind me what is the phenotype of Wnt5B?
5A.
That's what we did?
Yeah, 5A. We did the 5A.
Okay. I'm sorry. I don't remember the date. But anyway, the phenotypes are much milder when we delete any single ligand. We need to look at it more carefully, identify all the different ligands, hopefully in the future. Are there any more questions? Okay.
Okay, thank you so much. Thank you, Dr. Cha and Dr. Srinivasan, for that question and answer session. We're out of time, but if you have additional questions, please email NAVBO, email info@navbo.org, and we will make sure that we forward these questions to Dr. Srinivasan. I hope you found this research presentation beneficial. These webinars are brought to you by the NAVBO Education Committee. Our next webinar is Thursday, November 14th, 1 to 2 p.m. Eastern Time in the US with Hyung Chung from Yale University, entitled Endothelial Regulation of Metabolic Homeostasis. Look for the registration information in the NAVBO newsletter. And one more thing, please fill out the GoToWebinar evaluation form when you receive it in a subsequent email, and let us know what you think of the GoToWebinar format and any topics you may want to see in a future NAVBO webinar. Have everyone a good day.